And I believe we are live. It's always a bit of a gamble with this crowdcast thing, but I think we're good to go here. Hello, everyone. I see a lot of familiar names in the list, but if you're also new and joining us, welcome to the Escarpment Labs webinar series. We've been doing these for a few weeks now. Really appreciating the response that we're seeing. People checking in live, people watching afterward as well. We make this available on Crowdcast and then also long-term on YouTube. So if you do happen to miss any of our webinars, you should be able to access them here on Crowdcast or over on YouTube. It's been really awesome getting to record some of this stuff and share it a little bit wider. There's a lot of the kind of stuff that we often share on conferences and things like that, but it's really cool to have this sort of more democratic platform as well to share some of the things we do at Escarpment Labs. I've got a special guest here tonight. Is Neto is a microbiologist with a range of dynamic experience in food and beverage, as well as water quality, with a special interest in fermentation. She has a master's degree from Ryerson University, where she wrote an award-nominated thesis on environmental microbiology and toxicology. And during her time at Escarpment Labs, was the R&D biologist. She led projects that included improving strain-specific yeast health and performance. So some of that stuff that I was sharing in that yeast flavor diversity was a project spearheaded by Is. She also helped us develop some new QC methods for diastatic yeast and helped to develop new and optimized Lactobacillus souring blends. And that's what we're going to be talking about tonight. Currently, is the food safety and QA supervisor at Sunflower Kitchen in Toronto. And it's been fun catching up with you about that as well. I think a lot of us on the team at Escarpment Labs miss your presence, and we're still trying to figure out how we can go full steam ahead with R&D once the COVID situation starts to improve.
Yeah, I hope you manage. Thanks so much for the intro, Richard, and thanks so much for the opportunity to present this data. This was a really exciting project, I think, for all of us. There was so much information that we got from this, and yeah, I'm really happy to get the chance to tell you guys all about it. Yeah, without further ado, I'll start this presentation here. I'm going to go full screen, so I won't be able to see your comments until afterwards, and then Richard will take a Q&A after. So. Okay, so this presentation is based on a previous presentation that I gave at Holy Water. So I think there was a question earlier. If you did attend Holy Waters and saw my presentation, it will be similar, but I do have some new information there. So do stick around or go grab a drink and come back in a few minutes. I promise there is interesting new information. So here's Lactobacillus.
So I'm not seeing your screen. Sorry to interject.
Oh, I see.
Just make sure that we get that on screen. It's always one of the quirks of this platform.
Yes. Okay.
There we are. Okay.
How's that?
That is perfect. Thank you.
Okay, perfect. I guess I didn't click the right button.
Oh man, there's so many things that can go wrong.
Thankfully, we try to work it out before, and we know how to figure it. Well, at least we know how to problem solve here. Okay, so here's a little microscope picture of Lactobacillus with Anchorman ale cells for scale, I guess. I know if you're here, you probably already know quite a bit about Lactobacillus, and you're familiar with all the different microorganisms that are involved in making fermented beverages. But just to recap, we are going to talk about Lactobacillus. It's the picture on the right. And they're rod-shaped bacteria. They're known for converting sugars into lactic acid. They are ubiquitous in our world. They live in and on our bodies. They're good for our health and present in a lot of the food and drink that we consume. So here's what we based some of our research assumptions on. This is the general process for making a quick sour or kettle sour. There are lots of ways to incorporate different microorganisms in beer. For the purpose of the webinar, we're going to focus on the quick souring process using Lactobacillus. So what we mean by quick souring is the process of inoculating Lactobacillus into unhopped or low-hopped wort and then incubating it at an appropriate temperature for the lacto until it reaches our target pH, and then cooling it to yeast pitching temperature and then pitching the yeast. So there are lots of different ways to do this, and this really came out the first time I did this presentation. I asked people to come forward and share the ways that they make kettle sours, and there's lots of different ways that people do this. So I heard that some of you add a little bit of hops to the wort, some don't. Some of you pre-acidify the wort before pitching bacteria, some don't. Some of you keep constant temperature while you're acidifying. Some of you let the wort cool. Some of you purge with CO2. Some of you agitate the wort. Some sour in the kettle, and some have a dedicated FV. And souring time, we heard, can vary. Like we've heard some people say 12 hours. Some people say a whole weekend. Most people stop the souring process when the wort reaches target pH. And again, there are many ways of doing this. There's boiling, pasteurizing, partial boil, full boil, adding hops, etc. Another big difference here in terms of process and how people do things is the ideal temperature to pitch and incubate the lacto. So this is one of the main things that we're going to talk about today. And yeah, feel free to comment on the chat and share with everyone else participating what's your preferred method for making a quick sour and what temperature you use usually. So we wanted to start this project for a few reasons, and there's, as you can tell from there's so many ways of making kettle sours. There's a lot of anecdotal information out there for best practices, and really not that much in terms of empirical research and analysis. And another reason is that because this word of mouth on what strains work best for the process, a lot of people end up using the same or similar strains. And so when most people in the industry are using the same 3 strains, we end up reducing the flavor diversity of kettle sours. And as Carmen Labs, we wanted to work on creating a new blend as well, because we found some inconsistencies with the original blend when it was pitched at or above 40 degrees Celsius. So here's a summary of our method. We had agar plates, MRS agar, growing colonies of lacto. From the plates, we grew them in MRS broth, and then we propagated them in the Escarpment Labs lacto media, which is based on liquid malt extract. And then from there, we inoculated, we used this medium to inoculate 0 IBU worts at the same rate that we recommend people to inoculate lacto usually. So the same proportions. And we use these tubes, the 0 IBU worts, 50 mil Falcon tubes, in triplicate for each strain and at 4 different temperatures. And so we checked pH at 12, 18, 24, 36, and 48 hours. So a couple of notes, if you want to replicate this at home, which I'm assuming there's at least someone out there who really wants to do this, and you have time and you have some wild captures and probiotics, there are a couple of things to consider and remember from this project. There was some trial and error on the method, so this is a few things that I think is helpful if anybody wants to replicate this. Don't reuse your buffers. You can't sterilize your pH probe, so Lacto will definitely grow in the buffers and change the pH of the buffers, which I did confirm in the lab. And also, every pH check was taken aseptically. So if you have no way to follow aseptic techniques in your home, you can take one sample. Like your tubes will be good for only one check, and then it will be contaminated. And another thing to remember as well is that this entire project was made, it was tested on the same batch of wort. So first, to have comparability, we're using the same batch of wort and not and freezing it and using it again. So we're not using different wort for each test. And these are all incubated in a water bath. We use the Innova. I think we all really like it in the lab, the Innova sous vide cooker. Super accurate. Yeah, if you have any questions and you want to replicate this entire thing, which took us about 8 months, feel free to ask some questions. So here we go. Until we had 16 strains, 4 temperatures, 3 biological replicates. And this means 192 individual lacto ferments and 960 aseptic pH checks. That means a lot of 12-hour days, which is pretty fun. Here are all the strains that we used. So we have our code name, I guess, on the first column. That's kind of what I used during the project, as I was only referring to them by numbers. And then I think at some point during the project or at the end of the project, we got them sequence ID'd. So these are the sources for them. As you can see, there are a few commercial strains, lab strains, a few isolates, commercial isolates, and wild captures. Oops, what happened here? Yeah, so a few different strains. The species include Lacto brevis, Paracasei, Plantarum conchii, Delbrueckii, and Rhamnosus. So I have a few microscope pictures. They're not the most relevant here to this project, but I thought it would be fun to have them in the presentation just for everybody to see the morphological diversity of all these. And most importantly is that the take-home message is that even though they're the same species, the different strains can have morphological differences as well. So these are at 1,000 times magnification. And let's see here, these are the brevis strains. You can see the bacillus shape and the diplobacillus arrangement. So that means that there's 2 cells, usually 2 cells attached together. You can see more clearly here. That's pretty common for our strains of Lacto. Lacto Plantarum. This says 100 times, but this is with only the objective. So this is actually 1,000 times magnification. Yeah, so this is our Lactoplantarum commercial strain that we use in the lab. Yeah, here's another isolate that we use. And in here you see like this Lactoplantarum 652 was one that was isolated from a cavite culture. So you can see that their morphology is already starting to look a little different. Paracasei, again, a lot of them come in 2s, but sometimes you start to see here longer chains, 3s or 4s. And you can see here, this is also Paracasei. So this one looks very different. This was from isolated from a cavite culture. And here you start to see some much longer chains. That's the same like Lacto. And here's a common strain for Lacto sours, for quick sours. It's the Delbrueckii. I think a lot of you use it. Medium length chains. You can see that he's around 4 to 8 cells. And this one is Lactobacillus. This one was isolated from a thistle flower. It's actually a bee probiotic. It lives on bees, and it's quite a bit smaller than the others. Here's lactorhamnosus. So there's sort of connected multi-chains, which is interesting as well. So here are the results without further ado. This is kind of the overall picture of what all our 16 strains looks like at the different temperatures. I added the temperature in Fahrenheit next to the Celsius here in case anyone is connecting from the US. So yeah, the sort of blue-green is 30 degrees cooler, and then yellow-green 35, orange 40 Celsius, and dark red 45. So hotter is red and cooler is green. The x-axis here is time. Here in hours, this is the number of hours. And the y-axis is pH. So what we're looking at here for how we determine if a strain did well or not is if it drops pH fast over time. So at a quick glance, we can see that most strains acidified wort the best at the 35-degree incubation. That's the yellow-green lines here. Most strains did not acidify the wort at 45 degrees Celsius incubation. There's 2 strains, C209 here and 652. They also didn't do very well at 40 degrees, so they really worked best at cooler temperatures, like 30 and 35. As for individual differences, you can see with Lactobacillus, where is it? Delbrueckii soured equally well at 35 and 40 degrees, which tells us that the ideal temperature is between 35 and 40. Lactorhamnosus 295 was the only strain that acidified wort at similar rates across all 4 incubation temperatures, which tells us that this is a pretty robust strain with a wider temperature range than the others, which is good news for us. The Plantarum strains, you see Lpla, these 3 here, showed no change in terminal pH across different temperatures. So you see here, it doesn't matter at what temperature they and how fast they start, they kind of end up at the same point here. And what this tells us mainly is that optimal conditions for wort acidification are strain specific. So here's another visualization of terminal pH. And you can see pretty clearly that there are differences in terminal pH, where, for example, some of the Plantarum and Paracaceae strains are souring to the same pH regardless of temperature. So here's Lactoplantarum, our lab strains. Here, it doesn't matter at what temperature you incubate them with, they have a similar or the same terminal pH. And an interesting point here as well is that these are individual data points. So these are the triplicates. So there should be 12 points for each strain. If you don't see 12 points, it's because they're overlapping, which a lot of them are. And a side note, most of these triplicate measurements were very, very similar, if not identical, which if you've done any micro work, you know how unusual that is. They're quite similar. So, you know, when I started doing these pH checks, I thought they looked so similar. I thought there was something wrong, so I checked them 3-4 times each to make sure that you know, it wasn't too perfect. So this is actually they're behaving in a really similar way under consistent conditions, which really works in our favor when you think about it, when you're doing tests like these and thinking about the applications of the results as well. So here's a graph with our complete data, and this helps us look at the temperature dynamics for the 16 strains tested. And with the average here, we can see that the 35-degree incubation provided optimal results with the fastest temperature drop, and 30 degrees was second best. And 45 really didn't work for most of the strains. And this isn't to say that 45 degrees doesn't work for any lacto, because there are many commercially available strains that do perform optimally at the 40 to 45 degree range. But what our results indicate is that on average, if you're dealing with an unknown strain of lacto, it could be more likely that they are going to perform optimally closer to 35 degrees than 45. So in the end, again, this tells us that you have to know your strain that you're using. So here's the Hplc analysis, and this is a new slide in this presentation. And we have the results of the amount of lactic acid and acetic acid produced by each strain at each of the 4 different temperatures. So we kept the color the same. So if you remember, here are the colors. 30 degrees is blue-green, 35 is yellow-green, 40 is orange, 45 is dark red. So as expected, the lactic acid production correlates with the drop in pH. We see the highest lactic acid production at 35 degrees in Lactobacillus. Here, this one. Plantarum and KCI strains. So if you see the Plantarum and KCI strains, there's a high lactic acid production at 35 degrees. So this is lactoreminosis here. And if you remember for the previous slide, it's really consistent across all 4 different temperatures. And we also see here that it's producing a consistent amount of lactic acid at all temperatures tested. And it's slightly higher at 35 degrees. So moving on to the right side here for acetic acid, we see that the high acetic acid strains are the Lactobacillus strains. Here's another brevis strain, and Lactoconchia. See here. Acetic acid production was also highest at 35 degrees. So this is the ideal temperature for Lactobacillus and Conchia strains. All the others, they're not producing very much acetic acid at all. So we also did a sensory evaluation on these. We're not going to go into the results exactly in this webinar because it was really a lot of information. But I did include the fermentation information here because I think it's really interesting, and it's something that we observed during the first round of sensory ferments with Cali. So this was early on in the project, and this was before we had sent any samples for Hplc analysis. And for this step, what we did was we used 1 liter batches, again of the same wort, and we picked 8 different strains to sour them in duplicate at 35 degrees. And then we pitched Cali, added an airlock, and fermented at 20 C for 2 weeks. And what I want to show you here is that if you remember these, the Lacto brevis and Lacto conchii, these were the higher acetic acid strains. And they produce high acetic acid, which we now know that is the case for these strains after doing the HPLC. This correlated with a higher final gravity for fermentation with Cali. And this had a big impact on the perceived flavor of the final product. You can see here the final gravity, it's high, but let's remember this was 14 days. And these in particular were very, very slow. So the Plantarum, the other strains, they got to 10, 12 within about a week. But these two, after a week, this one was still 1026. Brevis was 1026, and Conchai was 1022. So they were very, very slow. And what's important to remember here is that like some lactose strains are heterofermentative, and they do make acetic acid. And we're pretty sure that this is why sometimes we see sluggish or stuck ferments with some kettle sours, especially if brevis strains are being used. Brevis strains are a popular strain. And this is especially true when there's a high concentration of lactic acid in the mix. I'm not going to get too much into the mechanism for why this happens in the cell, but as a summary, the cells bring in undissociated acetic acid molecules through the cell membrane. And once the molecules are inside the cell, that's when they lose a proton, and then they acidify the cytoplasm. And in the presence of ethanol, the proton pumps of the cell become saturated and overwhelmed, and it becomes harder and harder for the cell to regulate their internal pH. So if this is happening during the growth phase, what we see is slow and stuck fermentation. This is, I think, well researched in the wine industry, and you can look up wine industry research on this if you want more information on how this happens. So in the end, this maybe makes it sound like acetic acid is something we don't want, but really that's not what we're saying at all. And I had to say that for, eliminating acetic acid is really not the only answer. So when we ran sensory analysis on these samples later on at another round of sensory analysis, we did see that acetic acid can actually add a really nice character and depth to the flavor produced. I think this goes back to the flavor diversity I was talking about earlier as well. So as a follow-up to this, we did some tests to see if we could find yeast strains that are tolerant to acetic acid. And we did find a few, which is how we carried on the sensory component of this. And Scotland Labs, that there is a database with of the information on which strains are acetic acid tolerant, and we can Carbon Labs can make suggestions for which strains are better for using with acetic acid producing lacto, or suggest lacto blends that don't produce too much acetic acid that you can use with strains like Cali Ale and other strains that can't handle high amounts of acetic acid. There's many options. So this is the result of our first sensory analysis of the new blend. This is Lacto 2.0. And this table shows the pH drop comparing the original blend here on top, Plantarum brevis, and Lacto 2.0 blend on the bottom. And we did this at 35 and 45 degrees. and they all soured pretty fast and pretty well. So the original blend at 45 is a little bit slower, you can see here, and had a little bit higher terminal pH. You can see on the table. So once they reached terminal pH, we stopped the acidification with a partial boil and adding hops. So when we fermented with Cali, we can see that the fermentation, oh yeah, I was talking about the acidification earlier, sorry about that. So this is the acidification table and this is the fermentation table. So the fermentation was a little bit slower with the original blend incubated at 35. So this is Plantarum brevis at 35, which from the data that we now have from Hplc, we know that it's likely that this is where the most acetic acid is being produced out of these 4 treatments. So it's possible that this is what is slightly slowing down the Cali fermentation. So what this tells us is also for the variable results that we get with original blend, it can be potentially caused by different conditions and treatment and different temperature of souring, and that could be selecting for more growth of one strain over the other in the blend. In terms of flavor, we were really happy with the new blend. A lot of us picked up on some really nice tropical fruity notes like passion fruit that we really haven't seen before in single strain sours, like with Plantarum or with Delbrookia. So I was very happy with. So here's another sort of follow-up research question that we looked into after the Lacto research. So this project had sort of many tangents and many tweaks and many follow-up questions that we ultimately followed up on. This was one of the more interesting ones because early on I was testing the souring with the worts filled with those tubes. I don't know if you remember the Falcon tubes in the beginning. Some of them, part of the research, they were filled all the way to the top, 50 mils, and some of them halfway to 25. And when I noticed differences, I stopped right away and tried to see if there were any differences in the fill level, which is sort of a proxy for oxygenation and DO levels. And this is what we found. First of all, we got a DO meter and we tested the DO in the different fill volumes. And we for sure noticed that the difference in fill, the difference in headspace does mean a difference in wort DO. So we know this for sure. So we used this information to interpret our treatments as higher fill being lower DO and lower fill being higher DO. So 25 mils, only halfway fill, higher headspace, or larger headspace, higher DO, all the way to the top, smaller headspace, lower DO. And all of these results are after 24 hours. So if you see with some strains here after 24 hours, they have a low DO in the wort regardless of how much DO they started off with, which tells us that these strains could be taking up the oxygen. The brevis strains here are Kunkii. Oh, I circled them. Huh. I outsmarted myself. Okay, and some strains we see a really like a large difference in the amount of DO after 24 hours. So this tells us that the difference in starting DO is maintained after 24 hours. The higher DO correlated with slightly higher acetic acid in Lacto brevis and Lacto conchii, which, if you remember, they are the acetic acid producing strains. So with brevis and conchii, if they have more DO to start with, they're producing a little bit more acetic acid in the presence of oxygen. And they are, as we now know, the acetic acid producers. And the lower DO in Lactoplantarum means that there's a little bit higher lactic acid production. So if you have a lower starting DO, this means we ended up getting a little bit higher acetic acid production in Lactoplantarum strains, which is interesting as well to know how acid production reacts to DO in the wort. So take-home message, what does this mean for us? We now have a lot of information on how these strains behave and how much acid they produce and what temperature. And we know we have a little bit of starting information to look into impacts of DO in the wort. So we can use this to inform us and to help us make better decisions on strain selection and what blends to use and what combination with what yeast strains. And we can use this information also for process optimization now that we know which strains perform best at which temperatures and with how much or how little DO, which also allows us to dial in some flavor compounds. And from here, we can keep on expanding our strain-specific knowledge base, finding out more optimal conditions and treatments to achieve particular results and flavors, and again, expanding the flavor diversity. And we can test out some acetic acid producing strains with acetic acid tolerant yeast strains for maybe creating we talked about at Escarpment Labs was the idea of fast lambics, for example. The strains, like with conchii, is producing quite a lot of acetic acid. And we can also work on the creation of new blends for specific purposes, like, for example, making a blend with strains that are only isolated locally, or a blend with a higher terminal pH for people who like to leave their wort souring over the weekend and forget it and not over acidify, or more blends with higher flavor complexity, for example. And all this information is in the Escarpment Labs database. There's still a lot of strains to be tested. I only tested 16, and that took 8 months, so it's quite a bit of time. So there's a lot of applications to think about and test out, and this is really just the beginning. So we have the Escarpment Labs has 1,100 plus cultures, more than 50 lactose strains, and so the possibilities are really endless here. And this is the Escarpment Labs crew pre-social distancing. Everybody was happy. Good times. Thanks, everybody, and we'll be taking questions now. I'll stop sharing my screen if I can manage. Did I manage?
I think you managed.
Okay. Good.
All right. Excellent work. Thanks, Iz, for the fascinating presentation. Always exciting to share some of the work that's sort of behind the scenes at Escarpment Labs that ends up, when all things work out, ends up in some of these interesting fermentation products that brewers have access to that help sort of unlock and expand the flavor palette. And I think this is, one of those projects that really worked out well. And we're still sort of looking at, what else is in there for, some of those possibilities, like you mentioned, beyond just the sort of fruit forward Lacto 2.0. There's a lot of interesting stuff in there to sort of go on the opposite end and help to produce some of those more crazy sour beer flavors, like you find, Flanders Reds and Lambics and things like that in the future. So that's awesome. We have a number of questions. Before we dive into that, I do want to just mention everyone that we do have another webinar scheduled for this Thursday, so 2 days from now. We're going to be talking about hazy beers with Barncat Artisan Ales out of Cambridge, Ontario, not far from us. Jeremy from Barncat has been making this kind of beer basically since it's been a thing. He's an old homebrewing buddy of mine, and I remember him, bringing some of the first, Hill Farmstead, Alchemist, whatever beers, and then trying to reproduce them at home. So he's been in this game for a really long time. A lot of insight there. They make fantastic hazy hoppy beers. So excited to learn more from them on Thursday. But that's enough for our sort of future webinar spiel. We also have some more content coming up next week, 3 full days of content. So check our social media for more details on that. But there's going to be tons of stuff to come, after that as well. We've got a pretty good pipeline for you all. So if you don't mind, maybe it's a good time for us to get into some questions.
Let's do it. I should also say, I forgot to give a shout out to Caroline Tarawa, is that her last name, from University of Guelph, because she was the one who did the HPLC testing for us. Yep,
absolutely. And still plugging away. We collaborate with the University of Guelph and Caroline and George on all sorts of things, and that's resulted in a lot of really cool research.
So,
okay, our first question. This is a kind of funny one. The question is, does lacto flocculate? And if not, how does one bank it?
You can see there's some residue of cells in the bottom, but really not like yeast at all. So for banking questions, Richard, you probably have the better method here, or that's fine. Glycerol and.
Yeah, happy to answer. I think the answer here is that, well, for strain banking in general, you don't need a super dense culture. You just need something, and it needs to be alive. It needs to be in the right phase of growth. And then, you can freeze it down in glycerol, and that'll usually, as long as it's frozen cold enough, like we use minus 80, for example, that'll keep it safe for a very long time. Lacto, it doesn't flocculate, but because the cells are so tiny, you can pack a lot of cells into a small space without diffracting the light. So you can have something that it doesn't really look that dense, but it's actually got more cells in it than a yeast culture does, just because the actual particles of the little cells are smaller. So like even our own lacto cultures, oftentimes they're as dense as the yeast, but it doesn't look like it at all because the lacto cells themselves are so much smaller. So, you can take something that's basically as viscous as water, and you've still got, 1 billion cells per mil, and that's quite easy to then bank and store.
Yeah, and a lot of our, I think, denser cultures, the only way you can tell is that there's a slight swirly shimmer to them.
Yeah, the lacto shimmer. No one talks about that. We talk about that a lot, I think.
Yeah.
When you're training someone growing lacto, it's like, well, what does it look like? Is it shimmering or is it clumping? Yeah. Awesome. So our next question is there a large concern about producing botulism with lacto, and how can we go about minimizing this at home? Yeah, let's not give ourselves botulism right now.
I love this question because it's a food safety question. Botulism doesn't survive low pH. So in any cases of botulism that you get, it's usually from products that have a higher pH where botulism can survive. So I think if you open a food safety book and it's talking about botulism, the main example that they'll give you is carrot juice, because carrot juice has a very high pH, and also garlic, raw garlic in oil. All what these things have in common is a high pH. As long as you add an acid, you add vinegar, and you start to acidify, and the lacto really drops that pH, there is no risk of botulism. So the faster, if you want lacto action, the faster it starts to act, the lower the risk of botulism growing in your sample.
Absolutely. And for what I understand, the Clostridium botulinum is a little bit slower growing typically than lacto. So there's, especially if you're pitching a lacto culture, there's pretty low risk.
Exactly.
Yeah. Our next question, and this might be a tricky one to answer. The question is, does malt choice and thus nutrient levels play a big difference in lactic versus acetic production quantity?
Next project.
I guess not something really directly explored here. I think we can speak to the nutrient thing. So I think there were some experiments where we measured the FAN levels in the beer before and after, and if I recall, the lacto wasn't actually consuming a whole lot, right?
Yeah, I don't think there was much consumption, and we know from the sugars that lacto consumes at most maximum 1 Plato after 48 hours fermentation.
Yeah, we didn't show that, but I think that's a good one to mention. Is like lacto at most will consume 1 Plato, and it's mostly glucose and fructose. They're typically not touching any of the other wort sugars. The next question, and this is something that I think we have explored a fair bit in the past. Lacto and Kveik, pros and cons of co-pitching at a high temp versus traditional kettle souring.
So I think we actually did a co-pitch. Who was it that did? I think Jonah did a co-pitch in the lab that actually works really well, because they do lacto strains. Some lacto strains and Kveik strains can work really well between 35 and 40 degrees. So there is potential there to have a co-pitch kettle sour that is ready in 3 days, and this is one of the great things about it.
Yep. Yeah, I think that was Jonah who did that. And a few of us have sort of experimented with the idea. I love it. Like, pitch 35C and you've got fermented sour beer in a couple of days. We've sort of played around with optimizing that. Like, some of the Kveiks are just so hungry that they can actually slow down the lacto, and like maybe they are actually consuming the nutrients. So what we found works really well is just pitch the lacto, give it a 12 to 24 hour head start, and then pitch the Kveik, and then you'll because otherwise sometimes they were sort of stalling out in terms of acid production. But then if you give the lacto a head start, then you can get it nice and acidic and fermented out like super fast.
And I think we saw as well that most, if not all, of the Kveik strains that we tested are acetic acid tolerant.
They are, yeah, that's correct. The next question is if you've got a few-week-old or month-old lacto blend, should you make a starter or not make a starter? If so, what is the preferred method? Low-gravity DME, apple juice blend, etc.
That's, I think, a question for you, Richard.
I would say if it's older than 2 months, I would make a starter, simply because they don't survive super long, so you will see the viability drop over time in storage, just like with yeast. So it can help to make a starter. One thing to mention is lacto can grow to such high density that you often don't need to make a super big starter. So I would say for a homebrew batch, up to 500 mils would be really all you would ever really need. And there are some recipes out there. There's one where it's sort of like low gravity malt extract wort, like DME, mixed with some apple juice to drop the pH, but then also with some calcium carbonate added to buffer the pH, so the chalk. And apparently that works pretty well. That's not what we use in the lab, but that seems to be a pretty common approach on the homebrew side for people growing starters. And the cool thing about that is that the calcium carbonate will precipitate out, so you can sort of decant your lacto starter off of that buffer, and then it's not impacting the pH control of your actual beer. So that's kind of a cool method to sort of, because that is a key for growing lactose. You really need to buffer the pH so they don't just like drop the pH like crazy and conk themselves out. So apparently that works pretty well.
I also want to mention that for blends, something that we did see is that they have a variable survivability in the fridge, different strains. So it could be that if you have a blend, specifically for blends, that if you keep it in the fridge for a while, that one strain will survive better than the other. So if you want the specific results for that blend, you might not get what you're expecting after growing it and making a starter for pitching.
Mm-hmm. Yeah, that's a good point. Some of these things are a little hardier than others, even just between species.
For
like
sure.
The Plantarums are pretty bulletproof. The next question, I guess this is not necessarily lacto related, but sour beer or acid production adjacent. The question is, are you guys working with any of the Lactantia yeasts? And do you see a release of one in your future? I'm not sure if you got a chance to work with any lactic acid producing yeasts.
I have not.
Okay. There's a few we've come across with some of these projects where we're working with clients to pull out wild yeast from different environments, and some of the ones in the last year we've pulled out Lactantia, various Lactantia species, and these are sort of becoming infamous for being yeasts that can produce lactic acid, which is kind of cool because then you have something that can ferment out all of the wort sugars into alcohol and also make some sour beer acids as well. So there's sort of the potential there to have this sort of all-in-one, Swiss Army knife thing for sour beer production. And I know that there are a few of these on the market. Full disclosure, we haven't been super blown away by any of the Lactantias yet. There are a couple that we've pitched as part of those wild isolation projects for clients. We're going to have a webinar on that process in the future, by the way. There's a few we've pitched where we said, this is really cool. We've got this acid producing yeast. But the clients have ended up going with sort of more normal Saccharomyces yeasts. But, we are sort of, making sure that we're holding on to these things in case in case there's a chance to reuse them, because it is kind of cool to have a yeast with, something so radically different as lactic acid production, and have this yeast just be coming totally from the wild. This is so the next question is one that I think I would love to have an answer for, but I think we've both collectively struggled with a lot of the early steps of this. So the question is how to avoid THP in kettle soured beers.
Yeah, so I tried really hard to purposely produce THP in a sour and ultimately was not able to. So I don't know if anybody has a better process for compared to what we were using, but process wise, I don't know. Do you know this, Richard?
Yeah, if anyone has a way to just get THP in a beer without fail every single time, let us know, because that'd be very helpful for us to be able to actually study this, because it is a pretty fleeting thing, even in conditions where we think we'll reproduce it, like cooking Brett beers. Sometimes we can't get the flavor out. So, and then otherwise sometimes you're not trying to get THP and it shows up. It's total pain. But I recognize that this is a problem for flavor. It can be really distracting when you have this this flavor that shows up in the finish of a beer. And it'd be good to know more about it. And we do have a talk next week on THP, and there are some things known about it, like it does require certain amino acids as precursor to produce this off flavor. And then the other part is that it seems like that's linked to oxygen as well, where More oxygen can make it more of an issue. That being said, I think we tried this too, was, oxygenating sourwort, lacto cultures. We tried a lot of things to get THP flavor out, and it wasn't ever reproducible, which was frustrating.
Yeah. It could be that there's a threshold, or a couple of combinations of things that need to be present, that need to be there, that we still have to find out.
Yeah. This is a question that I don't think we can answer directly, but I think we will have an answer for pretty soon. The question is, does the relative difference between lactic and acetic production make an appreciable difference in the final ester profile?
Maybe, because, and I say maybe because I think the lactic acid, when we did the sensory, the lactic acid was associated a lot more with this lemony flavors. So I'm not sure about ester production. We didn't do GC analysis on this, I don't think. But for perceived fruity flavors, I think there's definitely some differences there to play around with, and they both can produce really nice flavors.
Mm-hmm. Yeah. The only thing I would add is just in general with yeast, if you've already got acid present, like lactic and acetic acid, that tends to reduce the ester production of the yeast. You tend to see lower esters. And for example, if you took the same yeast and fermented a kettle soured wort versus a standard wort, you'd probably see less esters in the kettle soured wort. So that may also be, support for some of these somewhat lower acid lactose to help support the yeast in making more flavor as well.
Lots of questions still. We need to do research, and every time we do this kind of research, there's more follow-up questions and more things that we want answered. So.
Yeah, that's how you know it's good research. If you have more questions than answers, that's good stuff.
For sure.
How necessary is pre-acidification?
It depends on what you want. It depends on your desired end results. For this project, nothing was pre-acidified, and the starting pH, at least for the sensory component, was a little high, and my thinking for that was, I was gonna make these sour beers for sensory evaluation, and I thought, if you pre-acidify, you're getting the acid that you're adding, and if you don't, you're making the bacteria work more, and they're gonna produce whatever other metabolites that are associated with bacteria growth. So there would be more flavor. For better or worse, you get more complexity and more flavor from making the bacteria work harder to reach terminal pH.
Yeah, it's always a balance, and I think we can also put this in the context of another question, which is how is Lacto 2 with regard to foam degradation? Are they prototrophic strains of lacto or oxytrophic? I can answer the second part.
Yeah, go for it, Richard.
Yeah, foam degradation in lacto, that's an interesting topic.
Yeah. Next project.
Yeah.
Add it to the list.
In general, it seems like pre-acidifying can help with foam. But there's also, as far as I know, hasn't really been a proper lab study on that. It's a lot of sort of anecdotes and experience. But that might impact flavor production as well if they don't if they're not breaking down those foam proteins. Maybe that changes the protein environment in the beer. But yeah, when we get into Lactobacillus and flavor in beer, this is very much new grounds. Unfortunately, it's not something that the sort of mainstream macro research world has really focused on. And in terms of craft beer, we're only now at the point where there's a lot of science happening.
Yeah, and like I said before, this is really just the beginning. This is just that first door opening up and answering a few questions and giving us even more questions to look into and try to answer and try to figure out.
For sure. Just one more question, I think, before we can wrap this up. The question was, have you put any research into finding or producing a hop-tolerant lacto?
Next project.
Yeah, I think 2 of the strains in this project were hop tolerant. We did screen them for the one of the hop resistance genes, the HORA, HORA, and 2 of them did have it. It was brevis strain and a Paracasei strain. So those are 2 species that are it's relatively common to pick up the hop resistance. Lacto brevis is pretty famous as a beer contaminant, or naturally present in a lot of sort of sour mixed cultures where there's some IBUs around, and same case for Paracasei. So some of them are that. Oh, go ahead.
I was going to say that was a PCR research from what I remember, and not actually like a performance testing.
That's correct. Yeah. So we were able to screen them for the genes, but we haven't been able to do any like plate-based like IBU assays yet. That was on the docket for this month, but then things happened, unfortunately.
Yep.
Can't help a global pandemic.
What can you do? We're all stuck at home.
What can you do? We can just sit at home and think about sour beer.
Yeah, and maybe make some and drink some.
Absolutely. Yeah. Excellent. Do you have anything else you want to add or you want to plug?
No, I think I'm happy that I got a chance to talk about this research again. Happy to answer questions anytime if anybody wants to bug me on social media. So.
Excellent. Well, thank you, Iz. Always
nice
to chat with you and talk about research. And I want to thank everyone for tuning in, listening to us talk about Lactobacillus and sour beer and all the work that sort of goes on behind the scenes to make the yeast and bacteria that we do. And really looking forward to what's coming down the pipeline. We're getting a chance to release a lot more of these sort of one-off or beta sort of batches through the homebrew limited editions, so watch out for those. 2 more came out this week, and we're going to try to keep those rolling out because it does seem like people are pretty excited about some of those ideas and products. And we might see some of the other lacto strains from this project get rolled into that. So just keep your eyes peeled for some of our new releases in the next few weeks.
Can't wait.
All right.
Yeah, thanks everybody for sending questions, and have a good night.
Cheers, everybody.